Hello, and welcome to Introduction to Bioinformatics Analysis, a course taught by Philip Richmond. This presentation will go over Section 1-1, Introduction to Course Structure, Computing, Bioinformatics, Sequencing, and Genomics. A quick foreword for this presentation is that this is not a comprehensive detailing of any of the subjects, not even close. This is simply to orient you and provide a useful base of knowledge that we will build upon throughout the class. The course structure for this class is going to be taught in the inverted classroom format, also called flipped classroom. The word cloud for inverted classroom will bring up things like student-centered, lecture replacement, peer instruction, blended learning. And really, the idea of the inverted classroom is that screencasts and slides for the lectures are going to be pre-recorded and made available to you so that you can watch them and go through them before coming to class. Within these screencasts, I will be going through examples of commands and tasks that you will actually be using in the assignment that we will do together in class. So then, when you come into class, there will be a short presentation followed by a problem set that we will work through together. The benefit of teaching in the inverted classroom format is that everyone is going to pick up this material at a different rate, and this way, if someone needs to go through the lectures multiple times before they're comfortable with the material, then they can do that. And then when we are in class, students will be able to help others that are moving at maybe a little bit of a slower pace. So together we are going to make it through this material. The course will be hosted on github.com. You should already have an account on GitHub, as it was a prerequisite for the course, but if you haven't, then it's very quick and easy to make one. So how about we go over to our internet browser, and if you go to github.com, then if you do not have an account yet, you can sign up for one here. Otherwise, we can click the sign in button. We will enter in our credentials, and then you're taken to your GitHub homepage. Now, GitHub is largely used as a version control system, which we will go through at a later time within the course. However, it is also where I'm hosting the course website. So we can find the course website by two ways. The first way is we go up to the search bar here and we type in Bioinformatics introductory analysis course. And when we search for that, it will bring up this course. And as you can see, the first thing at the top here is a number of different files. So as we go through the course, there will be different in-class assignments that can be found here, as well as one of the most important files, which is this README. And the README is presented here. You can see there's still a lot to do as I am recording this first video, but the idea is that in the README I have the course purpose, some general course information and prerequisites, as well as different resources that I will continue to add on, numerous different links, and then the course outline, where we are here currently in section 1, and you can see that there will be blue links indicating where the slides or the videos are. The slides will be entirely hosted on Google Drive, and the videos will be on YouTube. Another way to get to the course website is simply by typing in the entire URL, which is given here. And again, links for important websites are in blue, as well as the videos and slides, and hopefully everyone has done the prerequisites by this time. So computing via servers. In order to make use of a lot more compute power than you may have just on your own local machine, you are able to interact with what we call a server. And you interact with the server by entering commands in to your desktop or your Mac, your PC, and those commands will be given to what we call the head node or the controller node. Now, you can think of this as a master manager of the larger server system. Now, the manager does not actually perform the compute tasks that you need. Instead, it will distribute those tasks to the execution nodes. And this is where we make use of a scheduler. And so a scheduler, which we will go through how to use that at a later time, will take the tasks from the head node and distribute them across the necessary compute nodes. And all of this is linked together with a shared file system.
Compute Canada is an organization that works to bring computational resources to researchers across Canada. One branch of Compute Canada is called Westgrid, and they provide compute services to 4 different provinces, including BC, Alberta, Saskatchewan, and Manitoba. We can find out more information about Westgrid by going to www.westgrid.ca, which we can do now. So you've already been to the Westgrid website because you needed to register for an account, but we can find out more about the resources that are available to us by going to the Resources tab. In fact, the assignment for in-class work on Section 1 will require you to navigate this website, which we will be doing in class, so we'll not show any more of that here. A brief breakdown of the server system that's hosted at UBC for Westgrid is called Orsinus, or Orsinus. I actually have no idea how to pronounce this. I should probably figure that out. We're going to call it Orcinus for now. So the orcinus.westgrid.ca is going to be the way that we refer to this server. This is the address for the server. It is comprised of 384 nodes that have 8 processors and 12 gigabytes of RAM per node, and 544 larger nodes that have 12 processors and 24 gigabytes of RAM. And that brings us to a total of 9,600 cores or processors, which is a lot. Now, when we start using the server, we're going to need a couple different things. One is a way to interact with the head node, with the server itself, and we will do this using the terminal application on Mac or using MOBA Xterm on Windows. And then additionally, in order to get our jobs to run, we are going to need a way to interact with the different compute nodes. And this is done using a job scheduler, which we will interact with by submitting job scripts to the head node. A lot more on this in later sections. So bioinformatics, when I explain bioinformatics to people, I like to say that it is a combination of statistics, computer science, and biology, and at the heart of that lies bioinformatics, an intersection of these 3 different fields. Next generation sequencing, I could have an entire video about next generation sequencing, but there are lots of existing videos So what I recommend is that if you're not familiar with the Illumina sequencing platform or other sequencing platforms, the best way to get that information is to go to one of these two websites, which provide walkthroughs of how the technology is working, as well as lots of different videos on YouTube. This is a particularly good one from Illumina about how their technology works. So if you want to learn it, it's probably best to learn this from the people who develop this technology. I'm also more than happy to discuss the technology with you in person. Genomics, from Merriam-Webster Dictionary, is the branch of molecular biology concerned with the structure, function, evolution, and mapping of genomes. But I like this word cloud a little bit better. Genomics includes parts of bioinformatics, sequencing analysis, lots of different types of sequencing, looking at the RNA, transcriptomics, not sure what nanostring is, but there's that, CNV, copy number variants, PCR even is built into there. So genomics really is a large field concerned with genomes, is what I would say.
This course is designed for the Wine Research Center or the Wine Research Consortium here at UBC. And through this course, I will expose you to the tools and platforms on which you are available, or which are available to you in order to analyze yeast genomics data. Now, while the problem sets and examples will all be done using publicly available yeast data, the tools and techniques, as well as the platform, really is similar between different organisms. So if you are watching this and you work with human genomics data or mouse or fly, then these are still genomics tools that will be used in those different organisms. So the topics that I will cover in this course include resequencing and variant calling, genome assembly and comparative genomics, and RNA-seq differential expression analysis. That concludes the presentation for section 1-1.